Well, thank you so much to the Newborn Screening Translational Research Network for inviting me to share our story with you today. I'm going to share with you the impact that families can have on developing meaningful treatments for a disease called Angelman Syndrome, making the impossible possible. My name is Dr. Allyson Berent, and I am the Chief Science Officer for the Foundation for Angelman Syndrome Therapeutics. In addition, I'm the co-director of the Angelman Syndrome Biomarker and Outcome Measure Consortium, which is a pre-competitive consortium bringing together academics, clinicians, pharmaceutical companies, foundations, and families to talk about meaningful and measurable endpoints for clinical trials for Angelman syndrome. I'm also the co-director of something called InSync, the International Angelman Syndrome Research Council, which is a group of experts from around the world, most of which don't work in Angelman syndrome, in order to do a gap analysis yearly to help advance drug development and the entire ecosystem for Angelman syndrome. I'm the Chief Development Officer for a company called Biotherapeutics, and we're developing novel, innovative gene therapy and cell-based therapies for rare neurodevelopmental disorders. And I'm a veterinarian. I'm an internal medicine clinician and an interventional radiologist. I've been working in medical device development, designing and developing medical devices, and running clinical trials for the last 15 years. But the most important hat that I wear and the most important job that I have is I'm the mother to this beautiful little girl on the left. Her name is Quincy, and she lives with Angelman syndrome. In full disclosure, I was the co-founder and former chief operating officer of a company called GeneTx Biotherapeutics. And that company was recently acquired by Ultragenyx Pharmaceuticals, and that will make a lot of sense throughout this presentation. I am currently a consultant for Ultragenyx Pharmaceuticals, helping with their Angelman syndrome program. I'm a consultant and I advise many different drug development initiatives for numerous Angelman syndrome programs, as well as other rare disease organizations as a volunteer. And I'm also a member of the N of One Collaborative. So today I'm going to take you through a journey as a patient, as a caregiver, as a community member, as a member of industry, and then the holistic journey that brings all of these really important pieces and roles together in order to understand the best path for drug development for rare diseases. I'm going to start with a video, and this video is just a day in our life. This is a day in the life of my daughter, Quincy, and our family, and this is really a story behind how I got to you today.
When you meet her, the first thing you will notice is her smile and her laugh. You'll also notice that she can't talk with her mouth. She talks with an iPad.
I like them. I love them.
Quincy has so many strengths and so many challenges. If I ask her to say hi, she opens her mouth and says, "Huh." She has Angelman syndrome. It is a deletion of one of her genes, and it's totally random. I had a normal pregnancy with Quincy, but we started noticing things about her that were a little bit different. It was weird how unfussy she was. She never cried, and she was incredibly smiley. But she wasn't happy when she was looking at people. She was just happy looking at the fan. We were worried something is seriously wrong with her. So we asked if we can see a neurologist. The first words out of his mouth were, "Miss Berent, I have catastrophic news for you. What she has been diagnosed with is Angelman syndrome." I looked it up. What stuck with me the most was not walking, not talking, and having severe seizures. Under treatments, it said no treatments available. My head just started spinning. You know, she'll never go to college, she'll never go to school, she will never get married, she will never have kids. You know, all of the things you dream about for your beautiful newborn little girl, just taken away in a minute. And it was at that moment that I knew that I needed to educate myself and I needed to learn how to fix this. Because mommy and daddy fix everything. Bad news either paralyzes you or it motivates you. And for me, it completely motivated me to figure out a way to ensure that a treatment for Angelman syndrome gets outside of the mouse and into humans. the gene that Quincy is missing is actually present in all of us, including her, but it's silent. And all we would need to do is unsilence that copy of that gene, and that would be a mechanism in which we can actually cure this disorder. 2 weeks after she was diagnosed, a paper came out, and it was in adult mice with Angelman syndrome. And they gave these mice a drug called an antisenseoligonucleotide in their brain, and it reversed many of the symptoms that the mice had. And I realized at that moment that if they can do it in mice, why can't they do it in humans? How can we get this to my daughter? So in order to get this from mice to humans, it is incredibly expensive. It requires a tremendous amount of work, and it requires a tremendous amount of expertise. Most of the time, the steps between mouse and human are done through a pharmaceutical company. We didn't have that luxury. We had a concept, and we had a proof of concept and a mission with timelines that were not negotiable. And we had passion. And we were able to bring on the best people in the world, virtually, to help our company of 5 drive a drug to a human clinical trial that could impact children living with Angelman syndrome. Now, after all of this, the trial is open for enrollment, and I may be able to enroll my daughter in this study. This drug that we have worked so hard for could be in the brain of little kids. This is a miracle that I can't believe I'm a part of. My hope is that this is transformative. I want to see her life improve. I want her to tell me the things that she can't communicate in any other way. But I'm mostly thinking about who she is and who she might be. I just want her to say hi. That's what I want.
So when you get a diagnosis like this and you hear on the phone, I have catastrophic news for you, but your daughter has Angelman syndrome, the first thing you ask is, how do you spell that? And I'm a clinician, I understand medicine, and I had no idea what this man said to me on the phone. And I did everything every parent should never do, is I Googled it. And what I read were all the things that Quincy would never be able to do. She would never go to college. She would never have friends. She would never have children. She would never get married. She would likely not walk. She would likely not be independent. She would not self-feed. She would live a life of seizures, and she probably would sleep no more than 3 to 4 hours a night. And that is just devastating. Then I read that the prognosis for survival was great, and this was not deadly or degenerative. But I did know that her quality of life would be tremendously impacted. And then I had to think about, how do I give the best life and ensure that I can advocate for everything that she's capable of and ensure that she's accepted in this world that really isn't meant or designed for disability? How do I make sure she has the most fulfilled life? And I realized very quickly I had to advocate for Quincy, and I had to learn and execute if I could change the trajectory of her life than what I was reading that day. And about 30 seconds later, I went through this period of saying, I can't do this. And quite frankly, I don't really want to. I'm a veterinarian. I got to be exactly where I wanted to be in my career. I was working so hard in the area of research, clinical trials, and drug development, medical device development. I was really getting to the pinnacle of my career, and I had worked 18 years to get there. And how was I going to ensure that I gave her everything she was capable of and still have my professional career, as well as take care of the rest of my family? She had an older sister, and I was pregnant with her younger sister. And then I went through the guilt of, what kind of mother am I, that I care more about my own future and my own happiness than I do about caring for this child? How do we navigate this world of disability and manage a full-time life of therapy as well as this full-time job? But then I thought, there has to be a solution to this problem. Knowledge is power, and things are either possible or impossible, and there's no in between. And I had to figure out a way to ensure that finding a treatment for Quincy was possible. And I had to be brave, and I had to be smart. And so I learned everything I could about Angelman syndrome. It was a monogenetic, non-degenerative, random neurologic disorder. It had this very unique type of phenomenon called imprinting, where the maternal and the paternal copy of the 15th chromosome is read in every cell of the body except for neurons. And in neurons, the paternal copy is silenced by a long non-coding RNA called the UBE3A antisense transcript. And so in all of us, the paternal copy of this gene, the UBE3A gene, is not being read in the brain, in neurons, but it is being read everywhere else throughout the body, which is why this is a neurologic disease only. But in individuals with Angelman syndrome, they are not only having the silent copy of the paternal like you and I, but they're also missing the maternal copy, whether that's deleted or mutated, or some other types of more rare genotypes. And so by not having that maternal copy of the gene and a silent paternal copy of the gene, the symptoms of Angelman syndrome are quite profound. That includes a universal lack of speech. Most people with Angelman syndrome can't speak a single word verbally. They can have life-threatening and debilitating seizures, significant developmental delays, apraxia and dyspraxia, incoordination and ataxia. They have a unique happy demeanor, so they're incredibly joyous and happy children, as you saw in that video. But some kids can sleep 2 or 3 hours a night, which if you can only imagine what that does to a family and their caregivers who are trying to hold a job. They're unable to live independently throughout their entire life, both in childhood and into adulthood. They can have severe feeding issues, and there's no treatments for this disorder. So clearly a very significant clinical unmet need. One of the first papers I read when she was diagnosed literally saved my life. And that was this paper on somatic mosaicism in patients with Angelman syndrome. And what this paper showed was that as the mosaic individuals had more and more UBE3A being expressed, their symptoms and their clinical severity scale changed. For individuals that had peripheral expression of 1 to 5% of UBE3A, they had few to no seizures, they were ambulatory, they had some ataxia, and they had speech. For those that had 20% of UBE3A, they had no seizures, were ambulatory, and could speak in sentences. And those with over 40% of UBE3A were not actually reported in the database because they were considered asymptomatic, and likely neurotypical was considered above 40%. And so what does this mean? What this meant to me was that a very little bit can go a very long way, because that child with 1 to 5% of UBE3A expression is a different child than I live with. And I would do anything in the world to give that to Quincy. And so we knew that we didn't have to make it perfect. We just needed to make it a little bit better. So I had to behave like a clinician, and I had to understand what it was going to take in order to find treatments for her. I called every senior author I could on every publication, attended every scientific conference. And what was so incredibly clear was that it was the perfect decade to be a mouse with Angelman syndrome. But unfortunately, it was not quite the right decade to be a human with Angelman syndrome. Translational science was outside of the wheelhouse of most basic scientists, and basic science was outside of the wheelhouse of most clinicians who were doing everything they could for these kids. And so the valley of death was lying before us, where there was no ability to translate what was in an academic lab and bring it to our children. And that's where I had an idea based on my career of how to deal with that translational piece. So it was very clear that we couldn't wait for a treatment, but we needed to help shepherd one, and I had to behave like a clinician. So in October of 2015, I was lucky enough to be asked to join the board of directors of the largest research funding foundation in the world called FAST, the Foundation for Angelman Syndrome Therapeutics. And I became their chief science officer. FAST had put together a consortium of scientists from 6 academic labs around the country with a therapeutic focus, and they put that together well before my time in 2008. And they were really focused on developing treatments for Angelman syndrome. They were considering all therapeutic strategies and opportunities to scientifically de-risk each approach, because we know that each approach may benefit different populations of different ages and different genotypes at different times. And so I was tasked with creating a roadmap to a cure. We needed an ask to understand what it was going to cost to bring these incredible ideas to fruition. And it was at this time that we put a budget together for 6 therapeutic platforms to get to go and no-go decisions out of mouse candidate and into human candidate in order to bring them forward through IND enabling studies. And the cost of this was $5.8 million. And up until this point, we hadn't really raised more than about $500,000 a year. And so we made a commitment that we would do this in 24 months. We would have go and no-go decisions to get a human candidate into an IND. And that was our goal. So our roadmap for success really encounters 4 pillars. The first is that we can replace the missing maternal copy of the gene. We can activate the paternal silent copy of the gene. We can understand the biology of missing UBE3A and look at downstream therapeutics and address the synaptopathy of Angelman syndrome. And while we investigate all of this drug development, we need to be prepared for clinical trials. We need to characterize models, understand endpoints and biomarkers, be prepared for clinical trial execution, and understand the different genotype and phenotype correlations so we can understand how to test different genotypes so we can bring treatments forward for all individuals around the world living with Angelman syndrome. And so enzyme or gene replacement therapy is pillar number one, and we could do that through AAV gene delivery, hematopoietic stem cell gene therapy through lentivirus, autologous ex vivo gene therapy. Pillar number two is where we can activate the silent copy of the gene. We could do that through many ways. The first way that was shown was with topoisomerase inhibitors. The next way was antisense-oligonucleotides, which are currently in the clinic, artificial transcription factors, shRNA and miRNA for RNA interference, or CRISPR gene editing to knock down the RNA of the antisense transcript. And this was a CRISPR program for Angelman syndrome. And you could see the wild type on the left, Angelman in the middle, and Angelman after treatment with CRISPR editing. And this was treatment in an adult mouse model of Angelman syndrome. And what you can see here is that the Angelman syndrome mouse can be totally rescued as an adult after CRISPR gene editing and unsilencing the paternal copy of the gene. And so what this showed us was that age may not matter. Downstream therapeutics were also being carefully investigated, like ketone supplements, IGF-1 and IGF-2 ligands, brain-derived neurotropic factor analogs, and various different types of GABA allosteric modulators in order to improve the tonic inhibition in the brain of those living with Angelman syndrome. And so our pillar four is really the efforts that we're utilizing, supporting, and readying for clinical trials for innovative therapies. Not only are we developing all of the animal models for every genotype of Angelman, including the rat, the pig, and a large deletion mouse model, a UPD mouse model, as well as organoids, landing pads, and cell lines. We also have a natural history study that has been funded since 2006 that has 600 patients enrolled over a 15-year period. We have an Angelman syndrome global registry that has over 2,000 patients enrolled around the world. We have a fast global search and rescue initiative in order to find every patient around the world living with Angelman syndrome that's either non-identified or non-diagnosed or misdiagnosed. We have newborn screening efforts that are underway in order to ensure we have global patient identification, and we want centralized data access. We've developed a global center of excellence for clinical trial training and innovative therapy delivery for Angelman syndrome and other neurodevelopmental disorders, the FASTSearch Center for Translational Research, as well as developing the pre-competitive consortium, the Angelman Syndrome Biomarker and Outcome Measure Consortium. In 2006, we launched that consortium where 2 pharmaceutical companies were sitting at the table. 2 years later, there were 7, and today there are 31. What is most impacted in the lives of children living with Angelman syndrome and their caregivers to really understand minimal and meaningful change. And we've really been able to develop multiple endpoints in order to be able to best handle this heterogeneous population that's profoundly impacted where there's no qualified or validated endpoints for this population. And so we've created this dashboard of understanding all of the symptoms from communication, behavior, seizures, sleep, cognition, gross motor, and fine motor skills. And we've narrowed down all of the endpoints that have been evaluated in this population over the last 15 years to really prioritize the endpoints that would be included in clinical trials that are measurable and are meaningful with the symptoms of Angelman syndrome. And then in 2017, everything changed. What you could see here is a cell line of Angelman syndrome, where we have the blue line, which is showing the antisense knockdown, while the red line is showing paternal expression of UBE3A going up. And we had a human candidate for an antisense oligonucleotide that we had funded out of Texas A&M University. There was animal data that was out there since my daughter was diagnosed, showing the adult mouse model that had been rescued of phenotype. And the pharmacology here and the molecular biology showing that you can actually activate UBE3A in the brain using antisense oligonucleotide in a mouse model. And so what happened after this point was that our foundation decided to launch a company called GeneTx Biotherapeutics, and I became the chief operating officer of GeneTx. We formed a for-profit company out of our nonprofit in 2017 in collaboration with Texas A&M University, the foundation, and angel investors. We had a single focus, which was to develop this antisense oligo for the treatment of Angelman syndrome, understanding that we have unique genetic conservation with the non-human primate that we can use as our pharmacokinetic, pharmacodynamic, as well as toxicology model. We obtained orphan drug designation, rare pediatric designation, and fast track designation. And in 2019, before we filed our IND, we collaborated this program with Ultragenyx Pharmaceuticals, and they collaborated with us to have the option to acquire the company, and they helped us launch the phase 1/2 clinical trial in February of 2020 in the United States, UK, and Canada. In 2022, they acquired the company for $75 million, and that was just this past July. And that allowed the foundation to have an infusion of funds of over $50 million so that we can continue to advance all of our other shots on goal. And this was an incredible moment, not only for FAST, but for our entire community. What was more incredible was not the infusion of funds, but was the promising clinical data that encouraged this acquisition. I want to remind you, this was an open label study, and this was first in human. Changes reported objective and subjective data in both sleep, as you can see here. This was a change in sleep severity for individuals that were sleeping poorly. And you could see that the trajectory up shows a significant change in the severity of sleep in these individuals, a change in communication, both in expressive, receptive, and pragmatic communication. You could see the trajectory of each individual, which is rising over time. And there was a profound change in their communication abilities, as well as their global clinical impression of change over time in areas of sleep, behavior, communication, gross motor, and fine motor. You could see that there were a lot of 1+, 2+, and 3+ throughout this population, including individuals that were as young as 5 years of age and as old as 17 years of age. So we were able to see objective and subjective changes in sleep, expressive language, receptive language, fine motor, and gross motor skills. And so 8 years ago, when Quincy was diagnosed with Angelman syndrome, our therapeutic pipeline looked like this. There were 3 FAST-funded programs highlighted by those flags, and 5 overall programs that were being investigated in academic labs or with pharmaceutical companies. After we had that $5.8 million infusion of funds, 1 year later, you can see that that changed to 6 FAST-funded programs and 9 programs overall that were now moving into preclinical and drug discovery phases. 1 year later, you could see that we're in IND-enabling studies in a clinical trial, as well as multiple programs in drug discovery and preclinical with 10 FAST-funded programs, 14 of which were in the pipeline that were funded by others as well. 1 year ago, we had 34 funded programs, 12 of which were fully funded by FAST, 1 of which was in the clinic, this antisenseoligonucleotide, with 2 other ASOs in the clinic from other pharmaceutical sponsors, an IND that was submitted, and multiple FAST-funded programs that were approaching IND-enabling studies or in IND-enabling studies. And then this happened in May of 2022. The biotech industry took a crash, and that was incredibly unfortunate for many people in the rare disease space. Why? Because many programs, due to shifting economic times, as well as priorities and needs for really pulling in tight the reins of funding, our platforms went from 34 with 10 canceled programs down to 24. But what I want to highlight is that today we have 25 programs, 13 of which are FAST-funded. Not a single FAST-funded program slowed down or got canceled, and all of the canceled programs had nothing to do with programs that we were investing in and that we were pushing forward. And so what this really highlights is the power of patient communities and the power of fundraising that can advance programs and cannot allow things to slow down, because for FAST, Angelman syndrome will always be the priority. And so the chutes and ladders of drug development is incredibly powerful, and we can see things crash and burn regularly, but it should only crash and burn for bad science, not for changing priorities. And when FAST is funding those programs, that will never be the case. And so not only are we advancing drugs through the drug development ecosystem, but we need to advance all of the things it takes to get a drug into the clinic and to a drug approval. And that includes animal models, patient registries and natural history studies, disease concept models, endpoint development, newborn screening efforts, policy efforts, regulatory engagement, and diagnostic efforts. This is where we are today, and this is where we were 7 years ago. So what you can see is that we've made a huge change, and we've moved things forward in a tremendous way over the 7-year period. And some of our most proud advances this year have been our newborn screening efforts, as well as our clinical trial training centers for interventional and innovative therapies for rare neurodevelopmental disorders. And so the Angelman syndrome patient identification and newborn screening efforts is part of our initiative of the FASTSearch Rescue Initiative. Promising and transformative treatments are actively in trials, with 3 phase 3 ASO clinical trials approaching in the next 12 to 18 months, which could be over 300 individuals that will need to be enrolled over the next 12 to 18 months for these 3 phase 3 trials. really understanding the incidence as it's poorly documented and ranges between 1 in 8,000 and 1 in 164,000 is really important to understand. And there's tremendous benefit in patient identification, not only for community support for those families who are feeling alone and devastated by the developmental delay that their kids are experiencing, but also to ensure that they have access to standardization of care, access to clinical trials, and we're able to leverage our community army for progress, which is why FAST has had the success it has, was because of the families and caregivers who are so committed to advancing the ecosystem of drug development. Angelman syndrome supported newborn efforts right now are out of the University of Wisconsin-Madison, the Early Check out of RTI, and the Guardian newborn screening gene sequencing panel as well. 90% of our population can be identified using a simple methylation test. 10% of the population will require sequencing. The sensitivity and specificity for these tests are very high for blood spot methylation testing. And FAST is prepared to counsel and support these newly diagnosed families and ensure they have the resources they need to ensure they're avoiding difficult and debilitating diagnostic odysseys that we all had to go on, while providing mental and physical supports for both the today as well as for the tomorrow. Clinical trials are underway, and whereas dosing as low as 1 year of age in phase 1-2, now is the time to truly find these newborns with Angelman syndrome and ensure that their future is far brighter than those of us who have been living with this disease for years. So the Foundation for Angelman Syndrome also funded a $5 million initiative to advance clinical trial readiness and clinical trial centers around the world for Angelman syndrome, with our first one being out of Rush in Chicago, in order to be able to train other clinicians to deliver drugs that are innovative, difficult, like gene therapies and disease-modifying therapies. And our goal is to commit to 5 other centers around the world over the next 5 years so that we can ensure that patients are being treated regardless of their location. Not everyone has to travel to the very few sites in this country that are able to deliver these types of novel therapies. And so the Foundation for Angelman Syndrome Therapeutics does have an urgent mission to bring transformative treatments to our global community through discovery with academic collaborators, preclinical drug testing, developing endpoints that are meaningful and measurable, as well as bringing our community together in order to ensure that we have the right diagnosis at the right time and that there's the support that's needed for all families living with Angelman syndrome. And now we've added the clinical trial training centers so we can ensure that patients can be enrolled in clinical trials and we can bring access to everyone, not only around this country, but also around the world. And so our disease modification strategies, as I've already showed you, are pillar 1, with many different pharmaceutical companies truly invested in Angelman syndrome drug development. Pillar 2, unsilencing the paternal copy of that gene, with multiple different programs being invested in Angelman syndrome. And pillar number 3, downstream targeting, with multiple different pharmaceutical companies and academics invested in understanding the symptoms and how they can treat the symptoms as quickly and as safely as possible. But remember that one single drug to get approved throughout the entire life cycle of a single therapeutic platform can cost well over $130 million. And our foundation is sitting in this space right here, where we're putting in $10 to $20 million per platform, understanding that we're going to have plenty of failures along the way. But if we can remain the low-hanging fruit, that is why there are 25 programs currently being advanced for Angelman syndrome, where at the time my daughter was diagnosed, there were only 2. We have become the low-hanging fruit, and there will be many approved therapies over the next 5 years for Angelman syndrome. And I would like to thank our entire community of academic researchers, of pharmaceutical companies, clinicians, and of course our entire community for making that possible, to believe that this is a disease that is treatable and that we believe we can transform the lives of those that are currently born and those that are not yet born. So the holistic journey of drug development makes me acknowledge and be able to say honestly and truthfully that drug development is incredibly hard. But what I can tell you is that living with rare disease is far harder, because I have been lucky enough to do both. FAST works really hard to accelerate and de-risk all shots on goal, but it is very hard to rely on other parties to make your disorder a priority when times are tough and priorities change. But creativity in venture philanthropy has allowed FAST to continue to accelerate excellent science to ensure that we are pushing every single shot on goal, because the priority for FAST and the entire Angelman syndrome community will only change when our tagline changes. And our tagline is cureangelmansyndrome.org. And until that becomes curedangelmansyndrome.org, we're going to push as hard as we can, because we can't stop and we won't stop. So when we merge each focus of parent, clinician, foundation, and industry, that's the confluence of the best perspectives. The vision of our entire community has driven all of these programs to find an answer for all of the individuals around the world, which could be up to 500,000 living with Angelman. We just need to find them. And newborn screening is one approach that is going to help us really understand the incidence and really ensure that we can bring treatments to every child when they're born, and they don't have to live the devastation that the rest of us had. Infusion of funds has accelerated our ecosystem, but we still have a very long way to go. But we do believe we're going to have approved therapies in the next few years, and now's the time to really ensure we have the best screening possible. We have to work for every single one of these kids, again, those that are alive and those that are not yet born. So overall, by having a 15-year natural history study and the launch of the FASTSearch and Rescue Initiative, we are so incredibly excited to be able to work with MBS TRN to ensure that as many patients around the world as possible are found with Angelman syndrome, so we can work hard together in order to ensure that we're their rock and that we can find meaningful and transformative treatments for all of them and support them through every life's journey of their diagnosis of Angelman syndrome. So Quincy is my reason to fight every day for a change, because things are possible until someone shows me that they're not. And so far, everything looks possible. So thank you so much for your time today.